I'm Mark, and yeah, I'm sharing about EasyVitessce, which adds interactivity to these SCverse single cell and spatial biology plots. So this is a bit of the outline. I will describe the problem that we're solving, some background in case you're not aware of Vitessce, and then the where EasyVitessce fits in, and then the next steps. So the problem here is that when you're visualizing data from single cell or spatial experiments, or even more generally any data visualization, you typically are choosing between a static or an interactive plot, um, and you're not typically writing the same code to produce these different outputs. So it can be challenging if you have a static plot and you want to make it interactive. You're typically going to have to modify your code significantly and vice versa. But to solve this challenge, we don't need to invent new APIs. The key insight here is that ScanPy and Spatial Data Plot already provide intuitive plotting functions that people are using and know. So Vitessce, for a background, is a web-based framework for interactive visualization of spatial and single-cell data that the High Dive Lab is developing, and I'm leading in the lab. And this supports different SCverse data structures like N data, Mu data, or spatial data, as well as things like OME-TIFF and OME-ZAR, or even CSV. And it's very flexible. So you can see here on the right, in the HubMap data portal, we can visualize these data from these different types of experiments, so both spatial and non-spatial experiments, as well as these different multimodal assays. And we can view the spatial data in this layered approach, and even in 3D, as Eric and Eunice were showing. But being so flexible also has a trade-off in that it can be challenging for users to configure Vitessce for common use cases. So what EasyVitessce enables is to switch between the static and interactive plotting modes simply by importing this as a Python package. So you can see in this figure here that these are pretty much one line of plotting code. So for example, using a spatial data object, you can write onto your object.pl for the plotting module, and then one of these render functions like render images, and you can chain these things together so it's very concise to create a plot that looks like this for a xenium experiment, for example. So the static versions of the plots are on the left. These are what is currently produced by these plotting functions. And then on the right is a screenshot of the interactive version, and I'll show in more detail in the next slides as well. But simply by importing this package, you would get these interactive versions here in the right column of this figure. And so the EasyVitessce package supports a subset of functions from these different SCverse plotting modules. So for example, the heat map or the dot plot or the violin plot, or these different scatter plots from the ScanPy plotting module, as well as for spatial data plot, all four of the render methods that render different spatial elements within a spatial data object. And you can also disable the plots if you want to return to the static mode. You can disable the interactive plots. So one use case that this enables is to dynamically select a different gene of interest, or to select different cell types from using these controller components on the right that EasyVitessce will provide if you, for example, plot a violin plot. And then for things like a scatter plot, EasyVitessce even supports passing an array. If you're interested in multiple genes of interest, you can pass an array and you'll get this multi-view visualization. And since you're typically plotting the same data coordinates in these different plots, we link the zooming and panning interactions together. And then, of course, you can select alternative genes, and that will affect this final scatter plot in the list. Another use case for interactivity is to make some selection and then use the values in a downstream analysis. So here it's showing we're showing a scatter plot. And if you use the lasso selection, in the next code samples, you can obtain the cell IDs that you had selected, subset your and data object, and then make a new plot with this subset of data points. And then here we're just showing changing different visualization properties or parameters, like whether we're showing labels or how large the radius of the points are. So these are all the benefits of the interactivity that you can get here. Then this is showing you can then disable the plots and return. You can use the same line of code after disabling the plots to get back your static plot. So for a spatial example, we show that we can view this 10x Visium data set. And you see here this render images and then render shapes gives you the ability to show the spots in a layer above the histology image, and then you can again change different visualization properties. This gets also beneficial when you have these multi-channel images. You can toggle on and off different channels. This is a video of the previous example. You can change properties like the cell segmentation radius or stroke width. You can, as I said, toggle off the different channels and then change the window of the color intensity for each channel. So if you had done this with a static plot, you would have to keep making a new plot, change your code, and then replot with the new intensity window. But here you can just do it interactively. And so the next steps for this project are to have improved support for the points. So for Xenium and Mirrorfish to be able to modify the colors of points and filter the points based on a gene of interest. Right now, we just show the points with all one color, which is not super useful, but we're working on this. And other things to follow up on would be to combine multiple plots. So can we reuse the matplotlib API for generating these subplots for a multi-view visualization is an important question. And then how to deploy the resulting VUES visualization in a web app. So this is possible. You can embed VUES in a website, as you can see on these different data portals where you can use VUES. But a data analyst user might not know how logistically to go from their notebook into creating a website. So we can better document or have a tutorial for this use case that's common, of like you have a data set and then you want to send it to a collaborator or to your boss or so on. And you don't want to just send them a notebook, of course. So that would be, that's also something that we plan to work on. And that's it. And I forgot, this is how you can use EasyVitessce. So on vitessce.io, you can click this link in the navigation bar here to go to the EasyVitessce documentation, and we have example notebooks on GitHub, and you can install it with pip. You can also read about it on archive on this preprint, and we acknowledge the SCverse developers that made these APIs that we are reusing here, as well as the other members of the High Dive Lab. So thank you.
Thank you very much, Mark. And this is hopefully a tool that many in the community will find useful, because it's really a very low friction way of getting interactive visualization, where so far you might have been dealing with static plots that are, as Mark nicely demonstrated, quite limited in terms of what you can do when you're trying to explore a data set.